Hello everyone, my name is SYAKIRAH BINTI MAHADI, and today I will be presenting on the studies of human tear proteins. So tears are something that we experience frequently. For some people that might experience daily, like me, do you know that our tears contain hidden clues about our health? Yes, they actually do. So today I'll be discussing the fascinating topic of human tear studies, specifically looking into the protein found in tears and their implications for eye health. Our goal today is to explore how tear proteins can act as biomarkers for ocular health and help us understand the factors that influence the tear protein. So, what are tears composed of? Tears are not just water, they actually contain a complex mix of proteins, lipids, and other molecules. The key proteins in tear include immunoglobulins, lysozyme, lactoferrin, and lipocalin, which each playing different and unique roles. So despite their functions in our body, where are tear proteins mainly from? There are three sources of protein. There are three main sources of tears. First, the lacrimal gland, second, ocular surface epithelia, and lastly, the blood vessels in the conjunctiva. Next. So the lacrimal gland produces protein like immunoglobulin A, lysozyme, lactoferrin, and also lipocalin. These proteins actually help combat infections, maintain tear stability, and also manage lipid solubility for a healthy tear film. So next, in tear studies, these are recent findings in the tear studies. So samples are commonly collected using Schirmer strips, where a small paper strip is placed on the eye to soak up tears. So when these tears are collected, proteins are extracted and analyzed using advanced methods such as tandem mass specs and mass spectrometry. These methods can help us understand the protein composition and the relative abundance in the tear samples that we collected. So on the other hand, differences were found in the tear proteomes between Oregon and Florida, even when the tear volume was similar. So these differences suggest that climate and environment may influence tear protein composition. In this study, they also found that higher tear volumes led to greater protein recovery and variability, which means that not just climate and environment, but tear volume is also a key factor in tear composition. So next, why do we need to study tear protein? So studying tear proteins has promising applications in identifying biomarkers for diseases like dry eye syndrome, diabetes, and even Alzheimer's disease. So I give you an example for dry eye syndrome. So there was a study that explores how proteins in tears help diagnose and treat dry eye disease. So in the study, they discovered specific proteins such as the matrix metalloproteinases, MMPs, that are linked to tissue damage in dry eyes. So the inhibition of this protein can aid treatment. Inhibition using like doxycycline. So these findings is like they found the main source of the problem. So once they found the main source, they can find what can combat the problem. But how do we recognize these proteins, or as what we call biomarkers? So there are few techniques like proteomics. They use to identify these biomarkers. So how proteomics work is by identifying and analyzing the proteins present in a specific sample, like tear sample that we collected. So using proteomics, we can understand the specific role of the protein in health. Using these methods and this approach can provide a deeper understanding of biological system at the protein level, offering insights into both normal function and potential disorders. So in conclusion, next, in summary, human tear studies provide valuable insights into ocular health through non-invasive biomarker discovery. So with further research, tear analysis could become a powerful tool in diagnosing and understanding eye and systemic diseases. So that's all from me. Thank you so much for your attention.